In this video, we will present about Paramecium Caudatum, which is a species of unicellular protist in the phylum Ciliophoran or ciliate protozoan that commonly known with a shape resembling the sole of a shoe. We can identify Paramecium from the cilia covering the whole ledge of the cell. As we always see, Paramecium caudatum is a type of paramecium that lives in freshwater, ponds, pools, ditches, streams, lakes, reservoirs, and rivers. Before we discuss directly about Paramecium reproduction, let's try to understand its classification and structure.
Paramecium caudatum has two star-shaped contractile vacuoles and a cellular envelope densely studded with spindle-shaped extremosomes called trichosis. The species is very common and widespread in marine, brackish, and freshwater environments. Here are the main structures and organelles found in Paramecium caudatum. As an example is cilia, cytoplasm, nucleus, gullet, foot vacuoles, trichosis, and anal pore.
Paramecium classification is unicellular eukaryotic, so they are kept in the kingdom Protista. They are ciliatic protozoan and come under phylum Cetiophora.
The movement of Paramecium caudatum is facilitated by the presence of cilia, which are tiny hair-like structures that cover its entire surface. Here are some details about the movement of Paramecium caudatum, such as ciliary locomotion, avoidance response, contractile vacuole, orientation and directional change, and sensory structure.
For more specific, basically, a paramecium cell consists of two nuclei, which are micronucleus and macronucleus. The smaller nucleus is responsible for reproduction. In other words, it serves as the signaling organelle for asexual reproduction in Paramecium. When the environmental conditions are favorable, the smaller nucleus divides into two. This nucleus moves to either end of the cell. Following movement of the small nucleus to the opposite poles, the larger nucleus divides into two. After this, the cell divides transversely in the middle. The outcome is two similar Paramecium offspring, each having half of the cell organelles of the parent cell. Secondly, Paramecium also reproduces by sexual means, particularly when the cell is exposed to stressful conditions. For this process to take place, two Paramecium cells should come together.
The biochemical pattern of Paramecium caudatum, which is a single-cell organism, involves various metabolic processes necessary for its survival and growth. Here is a simplified overview of some major biochemical pathways in Paramecium caudatum. Firstly is glycolysis. Paramecium caudatum obtains energy through glycolysis, which occurs in cytoplasm. Glycolysis breaks down glucose into two molecules of pyruvate. Glycolysis also producing a small amount of ATP and NADH in the process. Next is photosynthesis. Paramecium caudatum is capable of performing photosynthesis due to the presence of symbiotic algae called zoochlorella. These algae reside within the Paramecium cytoplasm and use sunlight to convert carbon dioxide and water into glucose and oxygen.
In addition, the Krebs cycle, pyruvate produced from glycolysis into the mitochondria, where it is further broken down in a series of reactions called the citric acid cycle. This cycle generates more ATP NADH and FADH2 as well as releasing carbon dioxide. Meanwhile, Paramecium Caudatum is a single-cell organism, possesses a rudimentary electron transport chain etc. within its mitochondria, aiding in ATP production through aerobic respiration. This etc. involves a series of protein complex and molecules that facilitate electron transfer and ATP synthesis, enabling energy production for the organism function.
Next is protein synthesis. Paramecium Caudatum synthesis protein for various cellular processes including growth, repair, and enzyme production. This process involves transcription where DNA is transcribed into RNA and translation where RNA is translated into protein. Lastly, for digestion and nutrient assimilation, Paramecium Caudatum engulfs food particles into its oral groove and form a food vehicle. Inside the vehicle, enzymes break down the food into smaller molecules, which are then absorbed and assimilated into the cell for energy and other metabolic process. Now then, these are just a few examples of the biochemical pathway involved in Paramecium Caudatum cellular function. The organism's metabolism is highly complex and interlinked, enabling it to carry out essential process for its survival and reproduction.
First and foremost, in the article titled Integral Study of Paramecium Caudatum Acute and Chronic Toxicity, Sites of Injury and Distribution, Bioconcentration, and Body Burdens of Fine Metals. The authors make an analysis that this experiment would be helped to generate safer guidelines for protecting aquatic biota. To make this case a success, the 5 metals, which are copper, iron, lead, nickel, and mercury, were performed in the ciliate Paramecium Caudatum. For briefly, in terms of acute and body burdens, mercury was the most toxic metal while ferrum was the less toxic. Furthermore, the vacuole or irrigation chronic tests were more sensitive than growth inhibition chronic tests.
Secondly, in the article titled Paramecium Caudatum Enhanced Transmission and Effectivity of Mycobacterium marinum and Mycobacterium chelonae in zebrafish. In this demonstration, the author investigates if Paramecium Caudatum can serve as a vector of Mycobacterium marinum and M. chelonae by evaluating two larval, juvenile, and adult zebrafish. Moreover, infection was defined by histology observation of granulomas containing acid-fast bacteria in extraintestinal location, which is fish-fed Paramecium that containing mycobacteria became infected at a higher incidence than control. Therefore, they found that transmission was dramatically increased with the Paramecium vector compared to fish-fed bacteria alone. Overall, these results support that possibility of mycobacteria transmission in zebrafish facility to contaminate Paramecium culture.
Finally, the last case study that we choose is the article titled Interaction of Paramecium Caudatum and picornaviruses. Basically, the result of this research will be useful for management of reservoirs of picornaviruses in water and prevention of diseases. In this research, their data shows that Paramecium Caudatum were able to remove picornaviruses from solution in which the infusorians and viruses had been conjugated. In short term, no long term survival of picornaviruses in the presence of this Paramecium. But at the same time, the issue of the long-term presence of picornaviruses in Paramecium with minor release of viruses in the medium remain open, especially when Paramecium were destroyed by freeze or defreeze process.
Before we end the video, we hope that viewers got to learn something about Paramecium, especially Paramecium Caudatum, from 3 aspects that we emphasize, which are its basic interaction, the biosimilars pathway, and the case study. We sincerely apologize if input that we put in the video has a fault. Thank you for heeding your eyes and ear for watching, and see you next time.